G-protein coupled receptors. One type of signal transduction pathway that happens is through G-protein coupled receptors. First, the ligand bonds to the receptor that is binded to a G-protein and a GDP. This causes a shape change to occur and activate the molecule. The GDP falls off and is replaced by GTP, which contains more energy. The second messenger, cAMP, is produced, which causes an activation of protein kinase A in a cascade. The result is the activation to proteins that act as transcription factors in order to produce proteins to inhibit the response. G-protein coupled receptors. Related disease or disorder. CTX acts as a ligand and bonds to the G-protein coupled receptors, which causes very high levels of G-protein to be activated and cAMP to be released. This causes chloride ion channels to open and water and ions to be released into the intestines. Tyrosine kinase receptors. Insulin is the ligand that's created by transcription factors. Insulin bonds to the tyrosine kinase receptors, which causes phosphorylation to occur. The binding of ligands causes shape change in the proteins. This is a process called dimerization. In this process, the molecule IRS and kinases are activated. The receptors that contain kinases phosphorylate each other, bind proteins, and a response is activated. A series of molecules are also activated, and a vesicle containing GLUT4 is translocated. Glucose is allowed to enter the cell. Tyrosine kinase receptors. Related disease or disorder. As insulin is released into the body, the cell receptors aren't able to use the insulin signal correctly. An overproduction of insulin occurs. The RTK changes shape and the insulin isn't able to bond to the receptor. Transduction and response aren't able to occur. Growth factor signaling using RTK and G-protein signaling. Growth factors bond to RTK molecules that are attached to the cell membrane. A GRAS molecule is attached to those RTK molecules. The GRAS molecule moves along the cell membrane and bonds to adenylyl cyclase, where it then falls off to produce cyclic AMP. That cyclic AMP activates protein kinase, and that protein kinase activates a transcription factor. The transcription factor enters the nucleus of the cell and attaches to the DNA. The transcription factor causes the DNA to exit the cell and become RNA in the cytoplasm of the cell. The RNA produces cyclin with attached CDK. The cyclin and CDK then become activated, which then activates the cell cycle and mitosis, which leads to cell division. Growth factor signaling using RTK and G-protein signaling. Related disease or disorder. Growth factors bond to RTK molecules that are attached to the cell membrane. A GRAS molecule is attached to those RTK molecules. The GRAS molecule moves along the cell membrane and bonds to adenylyl cyclase, where it becomes hyper-RAS and remains bonded to adenylyl cyclase and produces an abundance of cyclic AMP, which is known as amplification. That cyclic AMP activates protein kinase, which is also produced in abundance, and that protein kinase activates an abundance of transcription factors. The transcription factors enters the nucleus of the cell and attaches to the DNA. The transcription factors causes the DNA to exit the cell and becomes a lot of RNA in the cytoplasm of the cell. The RNA produces an abundance of cyclin with attached CDK. The cyclin and CDK then become activated, and the cell cycle and mitosis begins, which leads to cell division.